Okay, we're going to look at DNA replication, but before we get into DNA replication, we need to understand the structure of a DNA molecule. How do we know the genetic code is found within that DNA molecule? And then what explorations led to the discovery of the structure? DNA stands for deoxyribonucleic acid. DNA is present in the nucleus of all eukaryotic cells. DNA controls the chemical changes which take place in the cells. The DNA tells the cells what they're responsible for producing, whether they're going to make muscle cells, blood cells, nerve cells, skin cells, etc. The DNA is also responsible for determining the type of organism that is going to be produced, whether it's a buttercup, a giraffe, herring, human. The DNA molecule, DNA, remember, is one of our macromolecules. It's an example of a nucleic acid. The subunit or the monomer for a nucleic acid was a nucleotide. The nucleotide, remember, has three parts: the phosphate group, the 5-carbon sugar, and a nitrogen base. When we look at the DNA nucleotide, again, we know we have that phosphate group. The 5-carbon sugar is going to be deoxyribose for DNA molecule. And then our nitrogen base. This diagram is showing us adenine, but remember that it could also be thymine, guanine, or cytosine in a DNA structure. DNA has a double helix. If you imagine this is a ladder, this is where you would put your hands on the ladder, and then going across, this is where you would put your feet going up and down the ladder. And this double helix, they've taken the ladder and it's twisted, and that's the double helix structure that you get. Where you would put your hands on the ladder, this is where you'd find your phosphate sugar on both sides. Phosphate sugar. And then coming off the sugar molecules are the nitrogen bases. Thymine goes with adenine, cytosine pairs with guanine, adenine again with thymine, we have thymine again with adenine. Those are the nitrogen bases: adenine, thymine, guanine, cytosine. Our nitrogen bases are grouped into two categories, and this is something that is done in organic chemistry. We have the purines and the pyrimidines. The purines are going to represent adenine and guanine, and you'll notice that the purines, they have two parts to their structure. The pyrimidines only have one part to their structure, and that's going to be thymine or cytosine. Again, looking at our DNA structure, imagine this is a ladder. It's laying on its side, leaning up against the side of the house. The backbone, again, you've got your sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's like where you would put your hands on the ladder. And then coming off the sugar is where we would find our nitrogen bases. Here's adenine. On the other side, we've got thymine. We've got thymine here. On the other side, adenine. Guanine pairing with cytosine. Cytosine pairing with guanine. They're bonded together by hydrogen bonds. Those red dotted lines in between those nitrogen bases are representing the hydrogen bond that's allowing for the nitrogen bases to combine together. Discovering DNA was a team effort. There were several scientists in different experiments that led to the DNA and knowing that it carries our genetic information. Beginning with Frederick Griffith. He asked, how is it possible that bacteria can make people sick? And through his experiment, he was able to find that it was through a process known as transformation. His experiment involved looking at bacteria being injected into mice. And what he had was a form of bacteria, but he had two different strains of it. He had a rough strain and then a smooth strain. And when he injected the mice with the rough strain, the mouse was able to survive. It lived. But when he injected the mouse with the smooth strain, he noticed that the mouse died. He then took that smooth strain and heat killed the smooth strain and injected the mouse, and again the mouse was able to survive. So then the idea was, let's take rough strain where mice can survive and heat killed smooth strain, again where mice survived, mix them together and inject the mice, but in this case the mouse died. And this is what led to the discovery of transformation, since there was a mixture of two different strands of bacteria, both individually allowing the mouse to live, but when they were mixed together, the mouse died. So this led to the idea that transformation was occurring, which means that one strand of bacteria is passing on genetic information to the other strand. That smooth strand originally killed the mouse. So something, even though they killed that smooth strand bacteria, something in the genetics had to be passed on to the rough strand in order for that mouse to die. Okay, Oswald Avery continued the study of Griffin's work. He wanted to know which molecule was important for the transformation. In his experiment, he was able to determine that it was the DNA that stores and transmits genetic information from one generation to the next. He took a sample of that lethal bacteria that was causing the mice to die. He then was able to go through, and separate out the individual components, the lipids, polysaccharides, proteins, and nucleic acids. He then removed one macromolecule at a time to see what would happen. He removed the lipids, did a culture. The culture showed it to be harmless. However, the mixture containing polysaccharides, proteins, and nucleic acids still showed the culture with a lethal substance. He then separated out the polysaccharides, did a culture of the polysaccharides, and it was harmless. But again, the mixture remaining with proteins and nucleic acids, which still show a culture that was lethal. He then removed the proteins from the sample, looked at a culture, it was harmless, but then the nucleic acids were left alone. That culture showed that it was lethal. Further experiments, he was actually able to separate out the RNA from the DNA and did cultures of those individually. RNA showed to be harmless, but the DNA continued to show a lethal substance. Therefore, that is helping scientists know that DNA is again responsible for the transmitting of genetic and storing of information, passing it from one generation to the next. Hershey and Chase continued with the discovery of proving that it was the DNA that was responsible for the genetics. They took a bacteriophage, which is kind of a virus that can infect a bacteria. And a bacteriophage has this DNA core, which is going to contain phosphorus, remember your phosphate group, and then it has a protein covering on the outside that contains sulfur. Let's look at the experiment. Hershey and Chase experiment, they took the bacteriophage, and in one case they were looking at the protein. On this side they're looking at the protein, and they allowed that bacteriophage to attach to the bacteria, and then they noticed that there was no sulfur detected in the cells at the end. Since there's no sulfur, they knew that the protein was not responsible for passing on genetic information. Then they looked at a bacteriophage. They were looking at the DNA core, which again is going to contain that phosphorus, allowed it to attach to the bacteria, and the bacteria became infected. They were able to detect phosphorus inside the cells, indicating that it's the DNA that's responsible for passing on that genetic information. Ernst-Chargaff, known as Chargaff's rule, tells us how the DNA nitrogen base pairs are going to pair together. Remember, adenine must go with thymine. A and T go together, apples in the tree. Guanine goes with cytosine, it pairs with cytosine. G and C, car in the garage. The amounts of the DNA molecule that will be present are going to be about the same. When we look at the base pairing for diagrams, this is showing us guanine bonding with cytosine. Again, those dotted lines connecting them, those are our hydrogen bonds. And then here's a thymine bonded together with adenine, again by hydrogen bonds. For Chargaff's rule, again, adenine bonds with thymine. Guanine is going to bond with cytosine. Again, imagine this, your DNA structure as a ladder. On the side, you've got your phosphate sugar, and then your nitrogen base comes off of that sugar, and they're going to bond, again, adenine with thymine. Don't try to memorize that you have adenine followed by cytosine, followed by guanine, followed by thymine. That changes. There isn't a set pattern for that. That's going to indicate more about our genetics, and we'll get into that in our next unit. What you guys need to know is that adenine is always going to bond with thymine, and cytosine is always going to bond with guanine. Rosalind Franklin used X-rays to determine that the DNA was a spiral shape. And then later Watson and Crick came along and said that they made the model for the DNA, the double helix model, which actually won a Nobel Prize. The last thing I'd like you to do is you're going to draw the complementary strands for the DNA nitrogen bases that I have given. I'm going to do the first one for you, and then you will do the last three. And this is a common question that you will see. You're pretty much guaranteed to see this on your EOC, where they'll say draw the complementary strand for the DNA, and they would give you nitrogen bases, and you would say cytosine is going to pair with guanine, adenine pairs with thymine, guanine pairs with cytosine, guanine pairs with cytosine, cytosine pairs with guanine, cytosine pairs with guanine, thymine is going to pair with adenine, adenine's going to pair with thymine, cytosine's going to pair with guanine. Now you guys do the remaining three, and we'll go over those in class tomorrow. Okay, we're going to look at chromosomes and DNA replication. This is going to explain how the genetic code is contained within our DNA. When we're looking at the DNA and the chromosomes, there's two types of cells. We have prokaryotic cells. These are single cells. They have circular chromosomes. This is representing the DNA, the chromosome material, just within the cell. And then we have eukaryotic cells, which contain a nucleus, and then within the nucleus we would find our DNA and chromosomes. The chromosome number varies between different species, in other words, between different organisms. Humans have 46 chromosomes. You guys need to know that. Mark it, highlight it, something. You need to know that humans contain 46 chromosomes. The giant sequoia tree contains 22 chromosomes. And then our little fly down here, he contains 8 chromosomes. So every species has a different number of chromosomes. When we look at the length of DNA, DNA is more than a meter in length, but it's still within the nucleus of a human cell. And so we wonder, how is that possible? Well, the DNA is going to coil and wrap around proteins tighter and tighter and tighter until it fits inside the nucleus. It's the chromatin and chromosomes that we actually find inside the nucleus, and we'll take a look at that. When we take a look at the chromosome structure, let me get my pen. Okay, this is representing the cell, and then you have within the cell the nucleus. They've pulled the nucleus out, and looking at the nucleus, then you have your nucleolus here. And then these X structures, which they're isolating one out, that's known as a chromosome. And if you uncoil the chromosome, you're going to start to see the chromatin, which is going to be the DNA material wrapping around proteins that are called histones. And if we continue to uncoil that, we'll eventually get to that double helix structure that we're familiar with for the DNA. And so within the nucleus, the DNA starts to coil around the proteins, making chromatin. Continue coiling, we'll get that chromosome structure, which again we find in the nucleus of the cell. The chromatin material is usually dispersed until mitosis. Now, mitosis is part of the cell division process, and our next unit will go through and talk about mitosis and the steps. But this is usually in the beginning of mitosis in prophase. This is usually when the chromatin is going to start to condense, coiling tighter and tighter so that we can see the chromosomes. As we take a look at DNA replication, let me get the pen, we need to look at the cell cycle. And this diagram is representing the cell cycle. The cell cycle, in other words, the whole life of the cell, we have a growth phase, which we'll call G1 phase, continuing through an S phase, which is known as synthesis. This is where DNA is replicated in the S or synthesis stage. And then we have our G2 phase, which is more growth, and then it's preparing for mitosis. So G1, S, and G2, those are known as the interphase. Interphase again is G1, S, and G2. This is where growth, DNA replication, and growth and preparing for mitosis occurs. For now, we're focusing on the S phase or synthesis phase where the DNA is replicated. As part of the cell cycle, the cell is going to divide to make more cells. But before the cell can divide, we need to ensure that the genetic information is passed on to the future cells. In order to do that, the DNA has to replicate first. If it doesn't replicate, then the new cells wouldn't have genetic information. So it's important that the DNA is replicated before cell division occurs. And we'll get into mitosis and cytokinesis in our next unit. Let me get my pen. Okay, each strand of the DNA double helix has all the needed information, and we need to be able to make a copy of that. So what happens when you have your DNA structure, that double helix, you'll have an enzyme that's going to come in and actually break apart the hydrogen bonds that were holding the nitrogen bases together. That enzyme's known as helicase. And then once helicase helps separate the double helix into individual strands, these are known as our parent strands, the parent being the original ones that we start with. Then you'll have another enzyme known as a primase that deposits primer. It puts primer down on that parent cell, so then a third enzyme can come in known as polymerase and help develop the new strands, the complementary pair. And then in the end, what you have are two identical strands of DNA, two double helixes that are identical to one another, and that is through the process of DNA replication. DNA replication, that literally means making a copy of the DNA, and that copy needs to be identical to each other, and then they have to be identical to the original strand. And these are the four enzymes that we're referencing. There are many more. We're just kind of simplifying this process for now. But again, you have your DNA. Helicase comes in and breaks the hydrogen bonds that are connecting those nitrogen bases so they can separate. This is showing it's separated. And then you'll have your primase comes in and it deposits a primer down on that parent strand, or the template as they're calling it here. Puts a primer down so that the polymerase knows what it needs to build for the complementary pair. And so your primase comes in and builds that new strand, or the complementary strand. And you'll notice that this complementary strand, okay, one is moving in this direction and one is moving in this direction. And that has to do with something known as 5' and 3', and we'll look at that in just a minute. Looking again at the DNA, again, you've got your helicase is going to help to unzips the DNA, separating it into two parent strands. And then you will have a primase that deposits a primer. And then polymerase is going to come in and make the new strand, depositing down the complementary pairs. So that in the end, we can have two identical DNA strands. The DNA polymerase, this is your DNA polymerase. It's another enzyme again. It's going to put a new nucleotide on the other side. It would bring, if you look at this nucleotide that's represented, it's going to put another nucleotide over here. I mean, we've got your nitrogen base, your sugar, and then your phosphate. It's building that new strand on the DNA. The polymerase is also responsible for proofreading. As it's building those new strands, the polymerase makes about 1 in 10,000 errors in the base pairing. Remember, adenine must go with thymine, guanine must go with cytosine. Then there are further enzymes that come through and they proofread it again, looking for mistakes. Once the enzymes proofread the polymerase, we're now looking at errors of 1 in 1 billion. All right, guys, remember when I said that on the diagram when I was showing you how the arrows moved in opposite directions? That's because DNA is anti-parallel, and you need to know this term, anti-parallel. The anti-parallel is the direction in which that new strand is made. On this particular structure that we're looking at for this segment of DNA, our new strand on the left would move down, and then this one on the right would move up. That's because the DNA polymerase moves from a 5' to a 3' direction. Again, from a 5' to a 3'. Now, understanding the 5' and 3', don't get too caught up in it. It has to do with numbering the sugar, the carbons within the sugar. And so at each one of these points on our sugar, there's a carbon. And the starting point is based on that first oxygen. And so if you'll look at your diagram, you'll notice our 3' is in the down position on the left, but on the right, it's on the top. Again, it has to do with numbering based on where the oxygen is located, where they start the numbering of carbons. You guys don't have to worry about that at this level, but what you do need to know is that the DNA polymerase, again, is going to move from a 5' to a 3', 5' to 3', and that's where we get the anti-parallel. One's going in one direction and the other goes in the opposite direction. Because it moves from a 5' to 3', one side is going to end up having fragments of our complementary pairs. And since there's fragments, we need to be able to connect those fragments on the DNA structure, and that's where ligase comes in. Ligase is going to glue those fragments together. DNA damage and repair. There are chemicals in ultraviolet radiation that damage the DNA in our body cells, and our body cells are known as somatic cells. Make sure y'all add that in your notes. Body cells and somatic cells are interchangeable because then we have sex cells that are different. The cells continuously need to be repaired. The DNA gets damaged and we have to repair that. The DNA polymerase and the DNA ligase replace and bond new nucleotides together in order to help keep this process going. Taking a look at DNA again for the replication, you have your 2 strands of DNA. Remember, helicase would come through, separate the strands, then your primase would deposit a primer, allowing for the DNA polymerase to come in and make the new strand, which we call a daughter strand. Daughter strand just means the new one. New strand means daughter strand. And then the original strand that was there at the beginning, that's known as our parental strands. We have the parent strand and the daughter strands.